All right, cool. This should be recording. Um, Howdy everyone. Nick again with a final exam practice test that I'm gonna run through with you guys. Same thing as the last one, 20 questions, 25 minutes. To emulate the, I guess, like structure of the final exam, I've included 6 questions from previous exams and 14 over the newer content that we've done, like the genetic stuff and the mitosis, meiosis, stuff like that. That won't be the exact spread of the final exam. I wasn't really told of that, but it should show you that the majority of the questions will be over newer material, and everything else like that we've learned throughout the semester will still be covered, but in lesser quantity. Um, yeah, let's do it. Let's do it. Gonna cast. Make sure that this works. Cool. So the first question, oh, I guess I should do a little disclaimer. This is purely for practice. Do not go into the exam thinking that these are going to show up. They are just so that you can get the pace down, get a feel for the type of questions you might see, and just overall practice. 20 questions, 25 minutes. Um, the final might be paced a little bit differently, but if you can handle the amount of questions in the amount of time that we see on the midterms, you should be good for the final. But yeah, let's do it. Let's do it. So question one, use the following map of 4 genes on a chromosome to answer the question. Which would you see the highest frequency of recombination between? And that's going to be A and G. We see the highest uh, Sorry about that. We see the highest frequency of recombination between genes that are furthest away on a chromosome. In this case, it's going to be gene A and gene G. This is because, like, during crossover, if we cross over at like certain sites between these genes, there's going to be a higher chance that they get cut between things that are further away because there's so much space between them to cross over at, right? On the other hand, if we look at the closest two, separated by these numbers or denoted by these numbers, meaning the distance between them, there's a lower chance that we receive a crossover event between genes that are closer together. So if this question were to say, what's the lowest frequency of recombination, that would be between W and E, which is conveniently not an answer choice. So uh, that makes it easier if you don't understand it for the exam, but you guys will all understand it. Anyways, G1 is associated with which of the following cellular events? That's going to be cell growth. During the G1 and G2 phases, we see just firstly, like regular cell activity on top of certain proteins and other signaling factors and cell structures being built in preparation for that mitotic event where it's separated. The cell is separated. Number 3, both cellulose and starch are polymers of glucose produced by plants, yet many animals can only digest starch. This is because what? So it's because that cellulose is made of beta glucose and requires symbiotic organisms to digest. Um, Starch and the most common form we see in that's made by like our bodies are made of alpha 1,4 and 1,6 glycosidic bonds, whereas cellulose is made of beta glycosidic bonds. These beta bonds, well, I guess it's not really important to know the structure of them, but they face a different orientation on that anomeric carbon, on the glucose, the sugar molecules that attach to other sugar molecules. And that orientation is super important because the orientation decides or is important to the enzymes that break down these sugars, these long sugar chains. So humans, with the enzymes that we produce through our genes, cannot break down beta glycosidic bonds, but luckily, some organisms have symbiotic organisms like bacteria in their gut that can break them down and then release those sugars to be absorbed in the digestive system of, like, I don't know, like cows, for example, they can eat grass. Yeah, so that's just how that works. Number four, inheritance patterns cannot always be explained by Mendel's models of inheritance. If a pair of homologous chromosomes fail to separate during meiosis one, select a choice that shows the chromosome number of four resulting gametes with respect to the normal haploid number n. So this is similar to one of the problem set questions that's talking about non-disjunction. Non-disjunction is the failed separation of chromosome pairs during mitosis, meiosis, either of them. And what we can see, we can draw a similar thing. Let's just do it only for meiosis one, or non-disjunction during meiosis one. If we see the normal number of individual chromosomes starting at meiosis, we have 92. Actually, let's do it in pairs because it's easier to see where it fails to separate. So we'll do 23 and 23. So if it fails to separate during meiosis one, during anaphase, which is when the chromosomes separate, we're going to see that the result is 22 and 24, right? And then whenever meiosis 2 happens, it's just going to work with what it has, right? So even though meiosis 2 might be working correctly, as in non-disjunction does not occur, it's still only going to be able to make whatever came out of, the non-disjunction event from meiosis 1. So what we see are four resulting gametes with an abnormal number of chromosomes. So what we see are these two right here are in -1, and these two over here are in +1. That is shown by answer choice D. I guess I'll go over if non-disjunction occurred during anaphase 2, during meiosis 2. If that were to happen, well, the original parent is still going to have the normal amount, and during anaphase 2 or anaphase 1, we're going to see a correct number. But if one of the meiosis 2 events occurs and experiences non-disjunction, we're going to see that one that's messed up. But the other one is probably going to be fine. You can't really tell, but this is what it would look like if only non-disjunction occurred right here during this separation. This is going to cause which answer choice? This one. That would cause C, but because we're talking about meiosis 1, D is the best answer here. Cool. Which of the following scenarios describes an example of epistasis? So epistasis is whenever one gene affects the phenotype or outcome of another one. For example, the answer choice has an example that is correct. Let's say fur color is indicated by a gene totally separate from the one that's being talked about in the question, right? So the dominant one would be green, or the recessive would be orange or something. A totally separate gene, the gene in question in this answer choice, can completely change whether it's green or whatever other color I said, even though it's technically not from that gene. It's a gene controlling another gene is the best way I can describe it. If that's confusing, search it up. Yeah, it shouldn't be too difficult to understand. But the C is correct answer for number 5. Cool. Clomipramine is a drug used to treat OCD, blah, blah, blah. So this is a question that you would find from the first exam. It's just a basic stoichiometry question, dimensional analysis question. There are multiple ways to do it, but how I would start is by taking the molecular weight of clomipramine, whatever, writing that down, and then just starting to take other numbers from the question stem and putting them in until we find the resulting weight of the drug that we need. So firstly, we'll plug in the millimolar. So we multiply that by 0.015 mole per liter. So no. That's just going to be 0.15 because millimolar, you go 3 to the left, right? And millimolar, that can just be moles per liter conversion. So I'm going to bust out my phone calculator. 400 times 0.15, that gives you 60. Because we have moles on top and moles on bottom, we can cross those out and we get a resulting number with the units grams per liter. Next thing we can do is cancel out liters by putting liters up here. Again, we're working with milliliters, so what we do is just move 1, 2, 3. That puts a 0 there and a decimal there. So we'd be multiplying by 0.05 liters over nothing. And that gives us 3. You can cross out the liters on top and on bottom. That just gives you grams over 1 or just grams. Luckily, that is answer choice D. If I messed that up, that would be really embarrassing, but I didn't. Cool. Number 7, if a cell has completed meiosis 1 and the first cytokinesis event and is just beginning meiosis 2, which of the following is an appropriate description of its genetic content? So it's going to have half the amount of DNA as the cell that began meiosis. That's because each of the resulting cells after meiosis 1 is going to have a pair of the homologous chromosomes. That's going to be half. And then after the second meiosis event, it's going to be half of the whatever we're talking about here. Cool. The mitotic spindle plays a critical role in which of the following processes? That's just the separation of sister chromatids. None of the other answers really make much sense. Mitotic spindle is just separating them, microtubular structure. Cool. Number 9 is a osmosis question. So yeah, on the left side of this little tube structure with a semi-porous membrane in the middle, we have a sucrose concentration of 2 molar, glucose concentration of 1 molar, sucrose concentrate on side B, we have sucrose 1 molar, glucose 2 molar. And it should be noted that it is this little semi-permeable membrane in the middle is permeable to both water and glucose, right? So off the jump, you should be able to just cross out the glucose on either side because the glucose is just going to move freely. It's not going to affect the water levels on each side. However, sucrose is not permeable. So in order to balance the concentrations of sucrose to water, you're going to have to move water around. And that's basically what we're going to do to answer this question. Right. So let's start with A and B right now. We have a sucrose concentration of 2 and a water level of, let's just put an arbitrary 1, right? Because the water levels are balanced right here. And then on side B, we have sucrose 1, water 1, right? Well, the final concentration of sucrose to water on side A right now is 2. And then on side B, it's just 1, right? Well, if we were to balance this without moving sucrose around, basically we'd want to increase the number in the denominator to lower the sucrose to water concentration. What I mean by that is moving water from this side to this side is going to decrease the concentration on side A of sucrose to water. So at the end, we might have something that looks like sucrose 2, water 2, sucrose 1, water 1. And we're kind of just making water out of nowhere if we're working with these discrete numbers, but these numbers don't really mean anything. They're just indicators of more or less relative to the other side. So yeah, basically moving water over to the left will decrease the concentration of sucroses to waters. And then that would give us a resulting balance in concentrations. And if we zoom out a little bit, the movement of water from the right to left is obviously going to increase the level that we see right here. So the resulting tube thing is going to look like that with the water. Like that. Cool. And the answer choice, the water level will be higher inside A than on side B. That's exactly what we see. Cool. Cinnabar eye color is a sex-linked recessive characteristic in fruit flies. If a female having cinnabar eyes is crossed with a wild-type male, what percentage of the F1 generation males will have cinnabar eyes? Right. So if cinnabar eye colors are sex-linked, and I think they're in this situation they'd be X-linked. That means that we have to set up a different kind of monohybrid test cross where we are working with the sex chromosomes, right? So these are from the male, these are from the female. And the female, it is firstly, the disease is recessive. So the female is going to have 2 copies of the cinnabar eye color as shown there. We can put just C to denote that it is the disease allele, or sorry, not disease, but like it is the cinnabar eye color allele. And then the male is wild type, meaning that it is what's most commonly found in the wild. It's not going to have cinnabar eyes. So we can leave these 2 chromosomes as they are, these 2 chromosomes, right? Cool. So let's do our test cross. Right. What percentage of the F1 males will have cinnabar eyes? Well, since we're working with males, we're only going to look at the ones with an X and a Y chromosome. And yeah, both of the males have a copy of the cinnabar gene, the cinnabar allele, right? That means that because males are hemizygotic, or yeah, I think that's the word for it. Meaning they only have one X chromosome, right? That means that whatever X chromosome copy they get, whether it's normal eye color or cinnabar eye color, that's just what they're going to be. So 100% of the males in this resulting generation are going to have that gene. That's what we see, the answer choice, 100%. Let me just make sure that it's still recording. Beautiful. Cool. Taxol is an anti-cancer drug extracted, blah, blah, blah. And it prevents microtubule depolymerization. Thus, Taxol stops mitosis by interfering with which of the following structures or processes. So microtubules is the biggest hint that's been given here. The depolymerization part is basically as microtubules elongate and contract, they go through like a kind of reciprocal movement called polymerization and depolymerization. Basically, all we need to know right now for this class is that the microtubules are involved with the mitotic spindle, which separates chromosomes. So if Taxol is preventing microtubule depolymerization, it's probably interfering with the mitotic spindle to stop cell division from happening. So mitotic spindle is the best answer there. Which of the following does not occur during the Calvin cycle? So the Calvin cycle, as we remember from exam 2 or 3, is the light reaction, Calvin. And these are working in conjunction basically to produce sugar molecules, right? Well, if we remember from the Calvin cycle, if I remember honestly, CO2 goes in. Yeah, because CO2 is fixed. Yeah. NADPH goes in and also ATP. And what comes out is NADP plus ADP and that G3P that goes into the sugar, right? So if you go back to our question, carbon fixation, that's what CO2 goes in for to be fixed. Regeneration of the CO2 acceptor, that happens during the regeneration phase. And oxidation of NADPH, that also happens NADPH from the light reaction is used to make those sugar molecules or the sugar precursors. What doesn't happen is release of oxygen. Cool. So yeah, definitely know the inputs and the outputs of all the biochemical reactions that we learn. Albinism is a recessive trait. A man and a woman both show normal pigmentation, but both have one parent who has albinism. Yeah, so if a man and a woman have the common pigmentation, but one parent had albinism, which is recessive, that means that we can make or both of the parents or both of the people had a parent who had albinism. So that means they're going to be heterozygotes, right? Because the parent is a homozygous recessive, and no matter what the other parent was, um, meaning that the other parent was a common skin color, common pigmentation. So they either had to be heterozygous, that's not heterozygous or homozygous dominant, and both of those produce only heterozygotes or another recessive, homozygous recessive. So if the two children that are mating here are normal pigmentation, that means they're still carriers. Super complicated, or not that complicated, but I kind of made it more complicated than it had to be. But basically, the man and the woman in the question are heterozygotes for albinism. So what is the probability that their first child will have albinism? Well, if we just do our test cross, the probability that the first child will have albinism is shown by the homozygous recessive. These two are carriers, and this one is straight up normal, does not have the allele for albinism. So the answer is 1/4. A man has extra digits. His wife and their daughter have the normal amount of digits. Couple second child has extra digits, right? So from this question, we know that the man is, let's use A. So he's this, the woman, the female, sorry, is not going to be homozygous dominant, even though she has the normal number of digits. We know that it's heterozygous because their second child has extra digits, right? So if she was homozygous dominant, they would only be able to produce heterozygotes, right? Which would all have the normal number of digits. However, because the question stem shows us that they had a second child with extra digits, we know that the female has to have the heterozygote genotype. Whoa, that's wrong. Boom. So this is our test cross. What is the probability that the third child will have extra digits? Well, doesn't matter if it's third, fourth, fifth, whatever. All of these events are independent of each other in the scope of this class. So the probability from this test cross that a child will have extra digits is just 2 out of 4, which is 50%, or 1/2. 15, which of the following processes, which of the following processes correctly describes alternative RNA splicing? It can allow the production of proteins of different sizes and functions from a single mRNA, right? So a pre-spliced mRNA might look like this. Right? And from cutting out these introns, you create your final gene, which is just this, right? But alternative splicing happens whenever certain regions, they may not be spliced in normal splicing, are spliced out, right? So we can have an event where this entire region between the red and the green genes, or sorry, the red and the green exons are cut out so that it produces this. And because this would produce a totally different mRNA, it would produce a totally different protein. Not totally different, but missing that one blue region in the middle. And that produces a protein of a different size and a function from the same original RNA. So that's what alternative splicing is. Pretty cool concept. Yeah. Marfan syndrome in humans is caused by an abnormality of the connective tissue protein fibrillin. And they're very tall and thin with long, spindly fingers, curvature of the spine, weakened arterial walls, stuff like that. Which of the following would you conclude about Marfan syndrome from this information? The answer is it is pleiotropic. Pleiotropic means that one allele, one gene, condition of the gene, basically expression, expresses itself in different phenotypic forms that are seemingly unrelated. For example, like height and arterial walls are both resulted. The conditions that result from Marfan syndrome, like the height that you get and the weakened arterial walls, both of those are a result of Marfan syndrome, and the gene associated with that. And that's the definition of pleiotropy. Cool. Measurements of the amount of DNA per nucleus was taken on a large number of cells from a growing fungus. The measure of DNA ranged from 3 to 6 picograms per nucleus. Which stage of the cell cycle did the nucleus contain 6 picograms of DNA? So this would be in G2. If we draw our chart, let's do G1, S, G2, mitosis. And then again, this would be G1. So in G1, we're going to see normal cell division, not cell division, replication of cellular components. Then during S phase, we're going to see a duplication. G2 is normal, mitosis, it divides, and then we're back at G1, the normal level of DNA. G2 is the correct answer because probably at every stage of G2, it's going to have the double DNA. But during S phase, you might hit it at a time where it's not fully duplicated to that 6 picograms. So G2 is the correct answer here. 18, a particular triplet of bases in the template strand of DNA is 5AGT3. What will be the corresponding codon for the mRNA that is transcribed? So let me just write it out again. AGT, 3' end. Basically, we know that if this is DNA and we want to make an RNA, what polymerase are we going to use? We're going to use an RNA polymerase that reads from 5 to 3 on the template. This produces a strand that is produced in the 3 to 5 prime direction. And of course, it's going to make the complement using ribonucleic acids. So that would be U, this would be C, this would be A, and the 5' end would be at the end. Right. And that's just your answer over here. Cool. Why are males... Okay, so the word was hemizygous, not hemizygotic. Probably really similar in definition, but I knew it was further on in the exam, and I wasn't sure if I got the word wrong. Anyways, why are males more often affected by sex-linked traits than females? It is because males, in the case of like humans and Drosophila or fruit flies, whatever, males are X and Y, right? So if we had a disease that was a recessive X-linked disease called XC, I guess, or that's the allele that messes it up. Even if we only got one copy of it as a male, you're going to express that disease. Whereas females, even if they only have one copy, this copy is going to be taken over. This one's going to be inactivated. And then you would probably have normal phenotypic conditions, right? So females, I'll put that up there. need two copies of a disease allele in order to express the disease phenotype if it's a normal like X-linked recessive disease, whereas males only need one. I'll put that down here. Males only need one copy. So, by chance, males are just going to have more disease conditions because they only need one of them. That's why we see that. They're hemizygous for the X chromosome, meaning they only have one X chromosome. Okay, which of the following statements correctly describes karyotype? So karyotype is just, you've probably seen a picture that looks like this, 1, 2, 3, 4, X, Y. And basically that's just a map of what all the chromosomes look like. Yeah, so the best answer choice here would be an organized image of the cell's chromosomes. That's what a karyotype is. Super simple. That is the exam. Thank you for watching. Hopefully the explanations are good enough. I think what will happen with my last sessions on whatever the date is, basically the Monday, the upcoming Monday, May the 1st, I'll just hold my session on Monday for anybody who might need further explanation of any of these questions. I'll probably also take questions on past problem sets or anything like that. But yeah, if you don't have any questions on this, you should be good to go for the final. Just keep studying, keep working hard, be confident, and yeah, knock it out of the park, finish strong, and then enjoy your summer. Thank you very much for watching. I think that's about it for this video.